We'll get started with the evening festivities. So what a great day we had today. Just tremendous platform presentations kept off by our industry partners. That was really nice. Nice day. So now we're into the next part of the day. And before I introduce our first speaker tonight, here's some housekeeping speeds from Donna. Those of you who haven't met Donna, where are you? Raise your hand. So most of you now met Donna. She's, we're gonna have a formal passing of the baton tomorrow morning from Liz to Donna, but you all know Donna now. So Donna gave me this note: right after our keynote, head to the upstairs back entrance where you were dropped off from the airport, and that's where the buses will run all night to and from our dinner. The last bus comes back from where we're going to be having our dinner tonight at 11:45. So I remember where they dropped us off. I don't remember if I know how to get there, but it's you know how to get there, Tony? No. Is it? Okay. All right. Well, that's enough housekeeping. So let's get started. And do we have the first slide? Great. So as many of you know, some of you may not be familiar with it, but a number of years ago, we started initially it was called MSG, now NMSG Fellowship Program. We do this in partnership with the American Brain Foundation, which is very important to Dr. Griggs. And it's a great program that our American Academy of Neurology, ABF, runs, and we basically cost share to fund what we call a fellow for 2 years. And so every year we have at any one time we'll have 2 fellows going. So Sophie, is this your first or second year? This is year 1. That's how. So we have them give a talk at year 1, which is not quite fair because they really have just started in their role as fellow. But most of our awardees are not true fellows. They're usually junior faculty who are at the beginning of their academic career, and so they actually have something to talk about already. So now, without further ado, then we will have Dr. Sophie Rengarajan. I did it right. Rengarajan, MD, PhD. Dr. Rengarajan went to Stanford, where she got her MD and PhD, and she's currently an instructor at UCLA, and we're going to hear about her research in Duchenne muscular dystrophy. So, Dr. Rengarajan, congratulations, and thank you for giving this speech tonight.
Thank you, Dr. Barron, for the very kind introduction, and thank you to the NMSG for inviting me to share our work today and also for funding me and supporting my career. I'm an adult neuromuscular physician, and I'm really excited about the novel genetic treatments that we have in our field, and I'm really curious to understand how these treatments alter the trajectory of patients with neuromuscular disease, and specifically Duchenne muscular dystrophy. So I think this group probably needs minimal introduction, but Duchenne muscular dystrophy is caused by loss of the protein dystrophin, which normally acts to stabilize muscle during contraction, and also acts as an anchor of the dystrophin-associated glycoprotein complex to the ECM. And typically in healthy muscle, sorry this is blurry, there's injury that triggers 3 waves of inflammation, and those occur in the context of muscle regeneration. However, in Duchenne, there's chronic muscle injury, and it's associated with asynchronous repair, improper resolution of that triggered inflammation, leading to ineffective regeneration, myofiber death, and replacement of muscle with fibrofatty tissue. And this leads to weakness and eventually premature death. Microdystrophin gene therapy seeks to fix the primary defect in Duchenne by restoring dystrophin at the sarcolemmal membrane. However, clinical trials of microdystrophin gene therapy have had limited success, and as Dr. Zaidman eloquently described, there are numerous adverse events associated with gene therapy. And so how can we think about making AAV-mediated gene therapy more effective and safe? So to give you a bit of context, muscle regeneration is typically triggered by injury, and in the context of injury, satellite cells, which are the stem cells of muscles, are activated. They proliferate and differentiate into muscle fibers. And there are numerous guide cells that function to help muscle regenerate properly. However, in Duchenne, these guides become activated and reprogrammed in order to drive inflammation and fibrosis, and that impairs typical regeneration and repair. So therefore, understanding the function of these guide cells in DMD pathogenesis is critical if we want to be able to entirely halt disease progression. And so to answer these questions, we perform needle muscle biopsy, and then on flash frozen muscle, we digest nuclei from muscle, and then perform single nucleus RNA sequencing. And this is a UMAP plot that was generated with my mentor labs, Dr. Carrie Michelli and Stan Nelson. So a UMAP is a way to visualize clusters of nuclei or cells that share similar genetic signatures. And so you can see that we can represent and resolve all the major cell types of muscle. And we can use this reference to understand how treatments alter the gene and cell signatures of DMD. So this reference set comprises 27 individuals with Duchenne and 5 individuals that are healthy. So we have been expanding this database to include individuals before and after various treatments, including gene therapy. And in a preliminary analysis of paired muscle biopsies from 3 individuals before and after gene therapy, we can see that the gene signature of myofibers actually more closely resembles the gene signature of healthy rather than Duchenne muscle fibers. And this is why gene therapy does work. This shows that we are improving dystrophic pathophysiology and increasing myogenesis. However, when we look at some of the guide cells in muscles, like the myeloid cells, we see that myeloid cells actually express a signature of trained innate immunity, or trained innate memory. And that trained signature, shown here in red in dystrophic muscle, actually persists even after gene therapy and does not resemble healthy myeloid cells. And when we look at satellite cells, you can actually see in healthy muscle, there's a population of quiescent satellite cells, whereas in Duchenne, the satellite cells proliferate and are activated, and we actually lose that quiescent population. And even after gene therapy, that quiescent population of satellite cells is not restored. And then we found a population of fibroblasts that exhibit an inflammatory gene signature. And actually, they very closely resemble a population of inflammatory fibroblasts that are seen in rheumatoid arthritis. And if we plot the proportion of inflammatory fibroblasts in muscle post-gene therapy against clinical improvement measured by a change in the percentile of NSAA before gene therapy and 1 or more years later, we see that there's a negative correlation between the amount of inflammatory fibroblasts and clinical improvement, suggesting that there potentially could be impairing the improvement that individuals could potentially achieve. And so our hypothesis is that inflammatory fibroblasts entrain an innate immune memory in non-contractile muscle cells, and this contributes to dystrophic pathology and is not resolved by gene therapy. And so for the remainder of the talk, I'm going to focus on endothelial cells, which are particularly well poised to contribute to both dystrophic pathology and also treatment response. So endothelial cells not only contribute to the delivery of oxygen and metabolites to muscle, but they also play an important role in maintaining the endothelial muscle barrier, muscle repair and regeneration, immune cell trafficking, and extravasation of a selective population of immune cells. And in Duchenne specifically, endothelial cells also play a critical role in maintaining chronic inflammation, promoting adverse events in gene therapy, such as thrombotic microangiopathy. The loss of nitric oxides signaling in Duchenne likely also contributes to tissue ischemia and myofiber injury. And actually, in a preclinical model of rheumatoid arthritis, inflammatory fibroblasts, similar to the ones I just showed you, can actually reprogram endothelial cells and impair their function. So in our reference atlas, we have over 13,000 endothelial nuclei that are resolved into common endothelial subtypes: lymphatic, venous, capillary, and arterial. And when we separate these nuclei based on healthy, shown in blue, and Duchenne, shown in orange, you can see that there's a pretty striking rightward shift of gene expression that's driven by Duchenne. And I quantified the proportions of each of these subtypes of endothelial cells and found that the proportion of venous endothelial cells is significantly increased, and the proportion of capillary endothelial cells is significantly decreased. And this is interesting because there's a phenomenon called capillary rarefaction that happens in many diseases and is actually driven by chronic inflammation, TGF-beta signaling, and reduction in pericytes, which can contribute to normal, healthy endothelium, suggesting maybe shared disease mechanisms of Duchenne and other immune-related diseases. And now this is a slightly different UMAP that shows a greater amount of nuclei. This is an expanded database, again showing blue healthy and orange Duchenne. And when we add in the post-gene therapy samples, which are comprised from 12 individuals post-gene therapy, you can see that in purple, there's a shift away from the dystrophic phenotype towards healthy. And when we quantify the proportions of venous and capillary, we see that whereas in dystrophic tissue, there's an increase in venous cells. That increase is actually suppressed in gene therapy significantly. And if we look at capillary endothelial cells, there's actually quite a bit of variability after gene therapy in the proportion of capillary cells across samples. So I next looked at the differential gene expression between untreated dystrophic muscle and healthy muscle, and I found that capillary cells exhibit the greatest number of genes that are differentially enriched. So I'm going to focus on those for the rest of the talk. And this is a heat map showing enrichment of pathways and capillaries of untreated DMD compared to healthy. And you can appreciate that in DMD, capillary cells contribute to adipogenesis, respond to cellular stress, contribute to interactions of immune cells, and chronic inflammation, all of which are and ECM matrix organization, all of which likely contribute to dystrophic pathology. And now when we look at the post-gene therapy gene expression, comparing it to healthy, we see that actually gene therapy reduces inflammation through TGF-beta signaling. We see less interactions with immune cells, but we still see evidence of ongoing pathology with adipogenesis and cellular response to stress that are both enriched. So then I compared post-gene therapy pathways, I compared the genes that were enriched after gene therapy to dystrophic muscle together, and I found that there's enrichment of pathways like myogenesis, VEGF signaling, and Notch signaling, all of which likely contribute to myogenesis and angiogenesis, highlighting a signature of repair. And also saw downregulation of TGF-beta and oxidative phosphorylation, which could suggest an improvement in metabolism of dystrophic muscle. But at the same time, after gene therapy, we also see enrichment in cellular stress, apoptosis, hypoxia, likely suggesting ongoing injury, and also platelet activation, complement, and coagulation, which could suggest adverse treatment events. And so to conclude, I showed you today that DMD causes reprogramming of the non-contractile muscle cells, and this promotes a hyperinflammatory state that persists after gene therapy. And in dystrophic muscle, we see a reduction of capillary cells and an expansion of venous cells, and that's partially and variably restored by gene therapy. Dystrophic capillary cells express the greatest shift in their gene expression and enrich pathways that are involved in fibrosis, inflammation, and adipogenesis. And after gene therapy, dystrophic capillary cells show enrichment of restorative pathways, but we still see some pathogenic pathways that persist. So I'm at the beginning of this fellowship, and I'm really excited about our future directions. So I only talked about capillary endothelial cells today, but I'm excited to look at the differential regulation of other types of endothelial cells and also understand how those endothelial cells interact within the dystrophic niche with other types of immune cells. And then all of this is based on transcriptomic data, so it's really exciting to be able to try to validate some of what we find in vivo. And as a teaser, these are endothelial cells that I isolated, cultured, and expanded from healthy dystrophic muscle. And this is an endothelial cell labeled with common endothelial markers and fluorescence. After validating those pathways, I think we can begin to start to think about what dystrophic signatures might predict a better response to gene therapy, and also think about how gene therapy and other types of treatments might interact together to really unleash the therapeutic potential of gene therapy. And we are continuing to increase our database of individuals and biopsies, and so we're very interested in recruiting more patients. We have open protocols and funding from PPMD to help with transportation, and so if you have patients that are interested, please refer them to us, and we're also happy to work with industry too. And so with that, I want to thank my mentors, Carrie Michelli and Stan Nelson. And this is a huge undertaking, especially production of the Alice that I showed you today. And so Kevin Chesmore, Florian Barthelemy, and Deidre Scripture Adams are postdocs who really were instrumental in generating and analyzing that data that I glossed over. I want to thank the remainder of the Nelson and Michelli labs, our collaborator and my mentor, Perry Shea. And then we're so grateful for the funding that has made this project possible, and I'm particularly grateful to the Neuromuscular Study Group for giving me the time to be able to do this research. And then finally, most importantly, I'm really grateful to the individuals and their families who contributed to and inspired this research. So, thank you.
Time for a couple questions. Dr. Damaczki? Okay. You're not going to make me run, are you?
So if I might ask the question about the fibroblasts, I was interested in that.
Yeah.
Given my interest in stem cells, I mean, they can be activated or resident fibroblasts, those inflammatory fibroblasts. Which type were they? And how do they compare to normal controls?
Yeah, that's a good question. So we think they're activated by DAMPs, damage-associated proteins that are released by probably injured muscle. We can talk more offline about this, but there are kind of a number of genes that are upregulated in them, and I can show you a signature. But the specific signature of those fibroblasts resembles a phenomenon in immunology called innate immune memory, and it's very similar to what we see in rheumatoid arthritis, which is exciting because it suggests that potentially therapeutics from rheumatoid arthritis could be transferable to Duchenne. And in fact, givinostat was first looked at in juvenile arthritis, so that's one of the treatments we're interested in looking at.
Yeah, Sophie, great talk. Thank you.
Thanks.
How soon after the gene therapy were the samples drawn? You said 3 patients, right?
Yeah, so the paired muscle samples that I showed you when I showed you the signatures in myeloid cells, fibroblasts, and satellite cells were from 3 individuals pre and post. And it is variable, so one of them was 8 weeks after gene therapy, another was 12 weeks, and a third was 1 year. So it is, yeah, we're currently in the process of biopsying patients at our center pre-gene therapy, 6 months after, and then for individuals who are started on givinostat, we're planning to biopsy them after givinostat. And this is actually a really well-tolerated procedure, and a lot of our patients are really interested to get this information and participate. Yes.
Okay, sorry. So I have 2 quick questions. The first question is that comparing Duchenne and healthy control, I mean, we know Duchenne have different stages, pre-symptomatic, at the more symptomatic stage and non-ambulatory.
Yeah.
So I'm curious about, the difference you see is at which stage?
Yeah, I actually have a slide I can show you. It's one of the hidden slides with the UMAPs. That's 4 panels. I don't have an exact answer to this question, but we actually have, so one of the benefits of the database that Carrie and Stan have created is that we really have well-curated patient information, and so we can go back and kind of understand how clinical information correlates with, and clinical course correlates with gene signatures. So I have a plot showing you that, actually there's a, yeah, that one is perfect. Thank you. Thanks, Gil. Basically, severity, gene expression seems to exist on a continuum. So here, healthy nuclei are shown in blue, severe are shown in red. Severe pathology, so nuclei from individuals who have a more severe dystrophic phenotype are shown in red. Green is mild, and yellow is typical. And so throughout actually most of our single nucleus sequencing data, we actually see that there is a spectrum that it correlates with severity of the disease. Yeah, thanks.
Yeah, Sophie, great talk. So Armando Vialta in our group has been looking at these fibroblast and fibroadeponogenic precursors. And we've identified these galectin-3 positive macrophages, and he's presenting some of this data at the World Muscle in a week or so. But it looks like there is this transition to these macrophages, which is supposed to be pro-inflammatory and regenerative, but they seem to be contributing to some of the scar formation.
Yeah, that's we're actually performing experiments where where I take supernatant from fibroblasts and actually use them to train myeloid cells. Our hypothesis is that it could be monocytes that enter muscle and are trained actually in the circulation or bone marrow, but it's possible they're trained in muscle as well.
Well, thank you so much, Dr. Ranjiro. Thank you very much. So Mike and I are going to do this together. I'll start. So now it's time for the annual Robert C. Griggs keynote presentation. So many of you have been here coming to this meeting for a long time. As we showed yesterday, Dr. Griggs started this organization in 1999. He stepped down as chair, we think it was 2016. And I remember then 2017 was the year that Mike and I took over as co-chairs. And both of us together and the leadership of the group were trying to decide what can we do to honor Dr. Griggs for everything he's done for this group, which he started, and for neuromuscular disease in general. What do you do for a man who has everything? And so what you do is you name an annual keynote lecture for him. And so these are the distinguished speakers that have been the Robert C. Griggs annual keynote presenters since 2017. And I know I talked to Dr. Griggs last week. This is the first meeting he's ever missed. He's really disappointed that he can't be here. I know especially for you, Valeria, since you're one of his mentees. But we're really proud for Dr. Hanna to introduce this year's Robert C. Griggs annual keynote speaker.
Great. So it's my pleasure and privilege to introduce our friend and colleague, Professor Valeria Sansoni, to do the Birch Griggs keynote lecture this year, a great honor. She is a full professor of neurology at the University of Milan. She, as many of you know, is the clinical and scientific director of the NEMO Center in Milan and has been since 2013. And over these past years, since she became the director, she's built an internationally leading, highly successful trial center for children and adults with neuromuscular diseases and has raised millions of euros for her research and trials programs. Just a bit more background on her. She trained in medicine and neurology in Milan. She did her PhD in myotonic dystrophy type 2. She did fellowships in Rochester, linking with Birch, as was said, and also in London. She's published hundreds of peer-reviewed research publications across many different areas of neuromuscular disease, but notably in myotonic dystrophy, in channelopathies, and also I think since she moved to Nemo in SMA and Duchenne. As many of you will know, she is an excellent clinical opinion, very clear, and she's also unusually an expert in child and adult neuromuscular disease practices across both. So it's really a great pleasure now to ask Valeria to give the Birch Griggs keynote lecture.
Okay. So my first question was, why am I giving this talk? So you've heard about all the famous people and the distinguished colleagues who discovered genes, published books and chapters, and revisited classification. So I thought another reason would be maybe because I'm retiring. So here's my first disclosure. I'm not retiring, and I'm not quite there yet. So I think that maybe what would be a good thing was to give just my recipe, my personal recipe of what's needed to enjoy your work and possibly add something to the community. So here's a list. And passion. So when I started to decide why would I get into medical school, I thought I would transfer my passion. I was at that time watching a TV series with Dr. Gannon, maybe because I liked him, I don't know, but he was a surgeon and he was saving lives throughout the whole series, and I thought I would never do that. Then there was a time where Rita Levi-Montalcini had discovered the growth factor, and I said, well, I will never do that. So the only thing I could do to transfer my passion was teaching. And preclinical helps because it gives you the approach. And the relationship with patients is nourishment. And so that's how I started my work. And then you need inspiration. And there are people whom you have to learn from. And here are the people that inspired me. So the first person was Giovanni Meola, whom showed me when I was a resident, not a resident, I was a student actually, that I was doing patch lump recordings on mice, and he was showing a cell culture of a Duchenne boy looking at acetylcholine receptors, and I thought, well, maybe humans are better than mice and I can do a bit more. So that's where I shifted, leaving a very easy career in basic science to get into clinical work. And then, here's Birch. First time I talked to him, he said, we have 15 minutes, you have 15 minutes, and you have to tell me what you want to do when you grow up. That was the first time ever I was meeting him. So time matters, and you have to be focused and know how to deal with time. And then he said, well, why are you publishing with the same names and the same people in your papers? I mean, you should do something, and it's your identity. It doesn't have to be sort of like a reflection of the people you're working with. And finally, and this is very unusual in Italy, if you are doing a work, you are the first name on it, and you are the one who gets credit for it. Then Rick Moxley. I have been running up and down stairs to the 5th floor of the neurological department at U of R. He kept talking and teaching me while he was running up and stairs. And the thing that I learned from him especially was patients and families first. And we went around Pennsylvania and the state of New York looking for the DM2 gene with him and Charles. And he brought donuts, very good donuts, to all the families. And I tried to work with him, but he didn't know me, and I sort of circled around him, and he said, well, no, there's other fellows before you. And then he gave me a chance because I showed him that I could attracted his interest. So again, I learned that you should give a chance to everybody, because if they insist and they really want to do something, maybe there's more behind it than just knowing them. And then, of course, who knows Dr. Moxley? Internal medicine is crucial. And so that was another thing I learned. And Charles. Charles only talked when he had something to say. And so I learned that I have not learned very much, but I'm getting there, that you should really keep quiet unless you really have something to say. And then he was very pragmatic and cautious. And when I came up to him like saying, well, you know, maybe thyroid problems are very common in myotonic dystrophy. They are associated with myotonic dystrophy. And he was simply saying, well, no, they're in the general population. You need more. And so there is where you learn and always be critical. And then in my last years, Eugenio Mercuri, I met him very late, 2017, and we started working very well together. And what I learned is you always have to think ahead. And again, that's my character. You have to learn to say things in a less direct way and try to get where you want with a nicer approach. And you need to collect data, that's very important. And we heard this several times during the meeting. Training and quality are mandatory, and I really got to a hold of that. And interaction with patient associations, foundations, and pharma are crucial for sustainability. So these are the people who really inspired me to get on with the work that I have been conducting so far. And one other thing is you have to learn to be focused. I shifted to a place where there were other diseases, but I think that you have to start with what you really know. And I had been working on channelopathies and myotonic disorders for a while. I won't say the number of years. And I was conducting basic science in the channelopathies, and then I ended up doing clinical work. And so it's a good thing to start where you're in your comfort zone, I would say. And then you have to also learn, at least that's, again, my recipe, to make the best of where you are. So at that time, I was in a cardiac hospital, and there was no point in trying to address other issues. And in fact, cardiac problems were one of the major causes of death in myotonic dystrophy. And so that's where you start. You start where your comfort zone is and where the environment helps you to bring up what you're working on. And then another thing is collaborations. So you have to try and collaborate within your hospital, with other teams in your own country, and with international colleagues you know and have been working with. And it's not nice when you are working on one field and there are national people in your country who are conducting multi-center studies and you are out. There's something wrong there. So critically, you have to sit back and think why that is and how you want to avoid that. And so one thing is you have to work with, well, you have to, that's my thinking, but Birch also said that. You have to work with the people you know and you enjoy working with and you trust them. And so this is how the collaboration with Nick Johnson and Jeff Statland, and Nick especially in myotonic dystrophy, is, I think, very successful, and we're having fun and we are working nicely. And so this is to show that from the international network, we're moving to the European network, and we're trying to coordinate the whole the different countries in Europe, and also in Italy with an initiative in the Italian Muscle Association. And then you need courage, because you need to understand what is it that you really want to do. So when I was in the lab, I was sort of jumping into something which I didn't know. So you have to really think that if you really want to do more, you have to take risks, and you have to just go where the flow goes, where you think you are going in the right direction. You may be wrong, but that's where I think you should do. That's what I think you should do. And these are the opportunities I have had, like I think many of you have. And one lesson that I took is you have to be happy after all and do what you like. And then you need awareness. And the site now that I'm working in, at the NEMO site, I thought that I was someone who knew about neuromuscular disorders, but when I moved to the NEMO site, I realized I basically knew only about myotonic dystrophy, and I had to restudy, rethink, and revisit, and one size does not fit all. And you also realize, because the NEMO site is a multidisciplinary and multi-professional site, that you have to be very humble and think that neurologists are not the savers of all that's around you in the field, and you need to sort of also give credit to the other professionals that are working with you. And again, bed to bench is increasingly important. And so this is a NEMO model, which you have heard, and this is how I then decided to sort of try and make this opportunity grow. And these are the big fives, and we are sticking to the big fives because I think, again, you have to set some limits because otherwise you will get out of your comfort zone, and you have to realize when you are ready to move forward in a different field or an additional field. And then you have to learn in a multi-center and learn from what's surrounding you. So having the opportunity of working with different diseases, and you realize that you can learn from those. And when I went to the NEMO site, I had so many ALS patients, and I thought, well, that's easy. We can focus on respiratory and nutrition and get what's done there to the other diseases. And so cross-fertilization is another important thing, but you have to learn how to set the limits there too, because respiratory is very different in ALS compared to myotonics or other diseases. We all know that in myotonic dystrophy, it's mainly a central problem, not a peripheral problem. Patients do not complain of lack of breath like ALS patients, so the indications for NIV, for instance, are very different. And if you apply the same criteria and you are not able to learn from other diseases, you are missing something. And so this is why then we set up to have different meetings and try to focus on respiratory, learning from ALS. And the same applied for nutrition. Same again for ALS. It's easy, or well, not easy, but it's easier to tell patients with ALS, so you should avoid eating this and that, because their lifespan is short and they know what's happening when they choke. But if you say the same things to a myotonic dystrophy patient, it will not work, and not even with a Duchenne patient and an SMA patient. So every disease is very different. I know I'm saying obvious things, but when you have the patients there, you realize how you have to tailor your interventions. And with with SMA again, we learned from the flow of the first treatments how to handle drug approval and the wave of therapeutic increase of patients coming in. And so again, that's a way to learn from others. And then you have to think ahead again and have some kind of vision of where are things going. And so you have to think as to what matters now, which is important. That's what we're doing with our job, but also what's next. And you always have to think about that. And so clinical trial units, of course, were one thing that was coming up. And observational studies were crucial. And you had to think that you had to obtain the data with quality and think of big data analysis and AI ahead of time. And again, think of some digital measures we've heard this morning. And I think this is something that you have to have an idea before that really is coming, really thinking before things are happening. And so you've seen this, but this is just to show that it took time. And even with AI, you have to start small. You have to start by doing some consultations, some remote rehab programs, using AI to deal with maybe some teaching and some instructions and some support for SMA patients. These are all very disabled patients who have used AI to create pictures to writings, pieces of poems and texts that they were writing with their eyes or with just one finger. And then you have to think digital, because that's where we're going, and you have to realize that before it's already there and you're not ready. And so with the Italian Neurological, thank you, with the Italian Neurological Society, we set up a digital neurology program to train 30 neurologists, young ones, to get into digital. And of course, I'm not a nerd person, I'm not a digital person, but I know, and I can think that, as many of us, of course, that that's where we should go. And so these are the modules. We set up a teaching course that's addressing AI into digital neurology. And then, of course, you need support. You need support from family, and friends. Because with friends, you always tell them, I'll be late. And the team. And again, you need to enjoy and have, that's the final piece of the recipe. Thank you.
Thank you, Valeria, for a really instructive talk. We do have well, actually, I don't think we do have any time, but we do have time for a couple of questions, or if anyone has any particular questions they'd like to ask about that approach to life and the recipe for success, which was really helpful. Okay. Okay.
So, in your now illustrious career, what's the biggest change you've seen in our field of neuromuscular disease that you think has been the most impactful?
So I think it's really this drive of treatments that are changing your perspective, and you are realizing what's important. And even today we heard, so who are you to say that this is not okay for your patients and it's not worth it? But we have to have the big picture, because if we are not setting limits, it's going to be hard. And the other thing is digital, because I really think that's going to change, not just in the analysis of the data for patients, but for us. We need to bring it into our practical clinic to allow us to work better, not to, as maybe somebody thinks we're losing our jobs, but to make our jobs better and give value to our jobs, because we don't have to waste time in putting and digitalizing and entering data and things like that. There are easier ways to do it, but we need to learn, because I'm not young, but I know that that's where we should go.
I did not expect that answer. Okay.
Okay, so now Rick is going to present the... No, you're still part of this. Present this.
Dr. Griggs would be very, very, very proud of you, and we all are, of course, but especially Dr. Griggs. So I want to award you the Robert C. Griggs Annual NMSG Lecture Award.
Thank you.
And thank you again for organizing this incredible meeting. So I guess now it's time to go find our buses. Yep. All right, thank you.